Hi everyone, my name is Chile.
My name's Ethan.
And my name is Jonathan.
Today we will gonna talk about seed dormancy. So first, we would like to introduce what is the dormancy. Seed dormancy is regarded as the failure of an intact viable seed to complete germination under favorable conditions. There are two types of seed dormancy. The first one is called enhanced dormancy, which means the embryo is constrained by its surrounding structures. We can see on this picture, this is the structure of the seed, and we can see testa here, which is also called the seed coat. When it is hard, the embryo cannot break and germinate, so it will stay dormant. Another dormancy is the embryo dormancy. It means the embryo is immature and requires a favorable conditions to reach a certain stage of development before germination occurs. What is the advantage of seed dormancy? I list some of it below. For example, the seed can pass through adverse conditions. It can reduce the competition for long distance dispersal and the storage of seed for later use. Ethan will talk more about it later. So how to break seed dormancy? It divides into two ways. First is the natural reduction. Second is the artificial reduction. For natural reduction, the dormancy stops when the embryo have a suitable environment, such as adaptive moisture and the temperature. But what is important that I'm gonna talk about is the artificial reduction. For example, people can apply some hydraulic pressure to weak the top seed coats, or using some machines to split the seed coats, exposing the seed artificially to heat, cold, or light. In this picture, we can see how a mature seed to germinate. A seed with favorable external and internal conditions, they will become mature seed and start their germination. But we also can give some artificial treatments to break the dormancy. Like scarification is to physically or chemically breaking down the hard outer coat of the seed. Stratification here is the incubation of seeds at a suitable lower temperature over a moist layer before conveying them to a temperature worthy of germination, like keeping them in lower temperature and moist environment, which helps them to germinate better in warmer condition. Exposure to white and the red light, hot water, growth hormones and chemicals. We all familiar with hormone, which is important, so I gonna record hormone from the lecture. What is important in seed dormancy is ABA and GA. ABA, which is abscisic acid, it helps seed development, cause dormancy, and control the stomata closure. Now for GA is a gibberellins, which will control the seed germination. So for active GAs, which is the GA3 oxidase. For inactive GAs, J2 will be the J2 oxidase. Jonathan will explain how they work in seed dormancy later.
Now we will go into why plants even bother going into dormancy. To redefine seed dormancy, it is when a plant embryo goes into a state of rest to allow it to survive extended periods of unfavorable conditions. These conditions can range from a variety of factors, which include things such as unfavorable weather conditions, the lack of or low levels of sunlight, and inadequate water levels. In this state, the plant will not grow unless certain conditions are met. These conditions include the proper amount of sunlight, temperature ranges that encourage growth, water, and of course, time. When these conditions are met and for a long enough amount of time, the plant can switch from dormancy to germination. A seed's ability to be dormant until favorable conditions are met allows the seed to grow in more optimal environments, where resources and space are more abundant. A seed that germinates too early can cause it to grow in an environment that's too competitive, leading the plant to struggle to grow or even die before reaching full maturity. For us, seed dormancy is a very important aspect of a plant's life. Seed dormancy allows us to store seeds in a dark and dry environment to use for later, such as when farmers store seeds for future seasons or when we need to consume them later on. Many of the foods we rely on in our everyday lives wouldn't be possible without seed dormancy. Corn, nuts, peas, beans, and wheat. All these products are thanks to seed dormancy. Without seed dormancy, we wouldn't have them. And finally, due to seeds' abilities to tolerate dormancy for long periods of time, We are able to preserve them against incremental and catastrophic loss of crop diversity in our ecosystems and environment, allowing us to rebuild any lost seed crop or low seed diversity areas when needed. These seeds are usually stored in seed vaults in colder climates, ensuring that these seeds will not break dormancy until we specifically want them to. The most famous seed vault being the Salval Global Seed Vault in Norway, which had more than 1.1 million seed varieties originating from almost every country in the world.
Next, we'll be talking about what are the major signaling factors that regulate seed dormancy. First, I'll be talking about ABA signaling factors, which include RCAR, PYR proteins. They are ABA receptors and they also bind to PP2Cs. SNRKs are also positive regulators, which are protein kinases. They regulate transcription factors along the pathway. Negative regulators also include PP2Cs, also known as protein phosphatase 2C. They interact with the C terminus of SNRK2s constantly, negatively regulating ABA signaling pathway. In this pathway, we can see under normal conditions and in the absence of ABA, RCAR, PYR proteins are inactive. This allows PP2Cs to repress the ABA signaling pathway by dephosphorylation and inactivation of SNRKs. Now, in the presence of ABA, the receptors RCAR, PYR proteins are now active. Consequently, this leads to the blocking of the activity of type 2C protein phosphatases. The lack of PP2C inhibitory activity causes the activation of SNRK2 protein kinases. This mechanism leads to phosphorylating downstream substrates, including the BZIP transcription factors, which activate ABA responsive gene expression. We can also see, in contrast, ABI1, also a subfamily of PP2C proteins, is a dominant mutation that allows the protein to be insensitive to ABA binding. This causes constitutive inactivation of SNRK2s. Secondly, we'll be talking about GA signaling factors. Positive regulators include the F-box proteins known as SLY1 or SNE as positive regulators of GA. They also form the SEF complexes. JID1 proteins are also known as positive regulators. They are GA receptors and increase the affinity of F-box proteins or DELA proteins. Negative regulators include DELA proteins, which activate negative regulators of GA responses downstream and also regulate upstream factors. We can see through this pathway, upon the activation of GA by GA20 oxidase and GA3 oxidase, the GA receptor of JID1 is able to now target the DELA proteins. This forms a JID1 GA DELA complex, allowing the SCF complex that contains either SLY1 or SNE to bind to and polyubiquinate the DELA proteins. The 26S proteasome then targets the DELA proteins for degradation. DELA proteins are not only important for GA regulation, but they also regulate upstream expressions of GA20 oxidase, GA3 oxidase, and JID1. Through this pathway, we can also see the DELA proteins regulate xerical expression, also known as ABA accumulation. This then shows how the GA signaling pathway and the inhibition of DELA proteins help balance GA and ABA expression. As an overview, we can see that these two pathways and their homeostatic interactions maintain seed dormancy. On the top left, we can see that dormancy induction is carried by ABA synthesis, which also leads to GA degradation, and ABA signaling to decrease seed sensitivity to the germination environment. On the other hand, we can see on the top right that the dormancy breaking is carried out by GA synthesis and ABA degradation. GA signaling causes germination. However, we can also see in the middle of the response that ABA is constantly cycling from a non-dormant to a dormant state, not allowing GA to negatively regulate seed dormancy without a strong GA expression.
These are our references. Thank you for listening to our presentation on seed dormancy.